welcome to the deep dive. We're here to cut through the noise, sift through the complex stuff, and really pull out those vital nuggets, hopefully giving you that aha moment without all the usual information overload. Today we're diving into something that, well, sounds like the opposite of life, really. Cell death. But hold on, we're not talking random decay here. We're focusing on a super precise kind of self-sacrificial process called apoptosis.
That's exactly right. And we're using a real cornerstone text here, Molecular Biology of the Cell, 7th edition, to get under the hood of how cells actually manage their own controlled demolition, you could say. It's not random at all. It's as basic to life as cells growing and dividing. We'll break down the molecular gears, look at the structures involved, and then, crucially, connect it all back to what it means for us, human health and disease.
Okay, let's tackle that paradox straight up. Why is something that seems so destructive, like cell death, actually so fundamental to, well, us being alive? Get this, in a healthy adult human, something like 1 million cells die by apoptosis. Every single second, 1 million. That just sounds unbelievably wasteful, doesn't it? Like, why throw so many away?
It does seem counterintuitive, yeah. But this normal programmed cell death, this cellular suicide, is absolutely essential, both for how we develop in the first place and for just keeping our tissues running smoothly day to day. Think about development. When you were an embryo, your hands and feet started out as these sort of paddle shapes.
Right.
The only reason you have individual fingers and toes now is because the cells between them were told to undergo apoptosis. They essentially carved out the digits. It's amazing.
Wow.
Or picture a tadpole losing its tail to become a frog. Same process, those tail cells get the signal for apoptosis. And in us adults, it's about balance. Cell death, specifically apoptosis, precisely balances out cell division. This keeps our tissues the right size, what we call tissue homeostasis. Take the liver, for instance. If it grows rapidly for some reason, maybe due to a drug, once that stimulus is gone, apoptosis ramps up to bring the liver back to its normal size. It's a constant regulation. And beyond that, it's quality control. Apoptosis gets rid of cells that are abnormal, in the wrong place, not working right, or even dangerous. Like immune cells that might attack our own body, they get eliminated via apoptosis.
Yeah, self-protection.
Exactly. Or cells with badly damaged DNA that could become cancerous. Often, apoptosis is triggered to take them out before they cause trouble.
That makes a lot of sense. So is all cell death this neat and tidy self-sacrifice? Or are there messier ways cells can go?
Good question. Big difference. What we're focused on, apoptosis, is very distinct from another major type, necrosis. In apoptosis, the cell shrinks, condenses, its internal skeleton collapses, the nucleus breaks down, DNA gets chopped up neatly. Crucially, the cell surface bulges out, forming these things called blebs, and the cell breaks into neat little membrane-wrapped packages called apoptotic bodies. And here's the really clever bit. The surface of these packages changes chemically. It basically puts up an eat me sign. This means neighboring cells, often immune cells called macrophages, gobble them up super fast before anything leaks out.
So no mess, no inflammation.
Precisely. It's incredibly clean. That's why even if loads of cells are dying by apoptosis in a tissue, you often don't see many actual dead cells lying around. They're cleared away so efficiently. Necrosis, on the other hand, especially the passive kind, is, well, messy. Cells tend to swell up and burst. Yeah, they spill their guts everywhere, and that usually triggers inflammation, which can cause further damage. There are some active forms of necrosis too, like necroptosis, but today we're really zeroing in on the precision of apoptosis.
Okay, this is where I find it gets really interesting. How does a cell actually make the decision? How does it initiate this precise internal suicide program? What's pulling the strings?
The absolute central players here are a family of enzymes called caspases. The name actually tells you what they do. They're proteases, meaning they cut proteins. And they're very specific. They cut target proteins right after a particular amino acid, aspartic acid, hence the asp and caspase. And they have a cysteine at their active site, the C. The key thing is these caspases are already present in almost all our cells, but they're inactive. They're like dormant soldiers waiting for the signal. We call these inactive forms pro-caspases. Now, there are two main types involved in apoptosis. First, you have the initiator, caspases. Think of them as the generals, like caspase 8 and caspase 9. They start the whole thing. They usually float around as single inactive molecules. But when an apoptotic signal comes in, it triggers the assembly of these larger protein platforms or complexes. These complexes gather multiple initiator procaspases together.
Bringing them close.
Exactly. Bringing them close allows them to activate each other. They basically pair up or dimerize, and then they snip themselves and each other into the active form. Once these initiators are active, their main job is to activate the second group. The executioner caspases. These are the soldiers carrying out the orders, names like caspase 3, caspase 6, caspase 7. Executioners also exist as inactive pairs, or dimers. They get activated when an initiator caspase cuts them at a specific spot.
So it sounds like once those initiators fire up, it's not just one activating one executioner. Does it amplify? It feel like this could be a point of no return?
You've absolutely nailed it. It's an amplifying caspase cascade. One active initiator can activate many executioner molecules, and then each active executioner can go on to chop up hundreds, maybe thousands of different target proteins in the cell. It creates this massive, rapid, and crucially irreversible wave of destruction. Once a cell truly commits down this pathway, there's really no turning back. And the targets of these executioners are very specific things that lead to the cell's demise. For example, they chop up the proteins that support the nuclear envelope, the nuclear lamins. That causes the nucleus to break down. They also cleave an inhibitor protein that normally keeps a DNA cutting enzyme, called CAD, locked down. When the inhibitor is cut, CAD gets unleashed and starts slicing up the cell's DNA into those characteristic fragments you can see on a gel.
That DNA ladder pattern you sometimes see in labs.
That's the one. That's a hallmark of apoptosis. The executioners also dismantle the cell's internal structure, the cytoskeleton, which leads to that membrane blebbing we mentioned. They cut proteins that hold the cell to its neighbors, allowing it to round up and detach, making it easier to engulf. And importantly, they trigger changes in the cell membrane itself, flipping certain lipids to the outside to create that eat me signal we'll come back to.
Such precision. Okay, so where do those first signals come from? How do the initiator caspases get that initial nudge? You mentioned different ways this can start.
That's right. There are two main routes, two main pathways to activate those initiator caspases. The extrinsic pathway and the intrinsic pathway. The extrinsic pathway is triggered, as the name suggests, by signals from outside the cell. It involves specific receptors on the cell surface called death receptors. Imagine, say, a killer lymphocyte, an immune cell, recognizing a target cell that needs to go, maybe it's infected or cancerous. This killer cell displays a molecule, like Fas ligand, on its surface.
Like a death warrant.
Sort of, yeah. It binds to the corresponding death receptor, Fas receptor, on the target cell. This binding pulls several receptor molecules together. This clustering exposes parts of the receptor inside the cell, which then recruit adapter proteins. These adapters then grab hold of inactive initiator caspases, specifically procaspase 8 in this case. This whole assembly receptor adapter procaspase forms a big structure called the DISC, the death-inducing signaling complex. And inside the DISC, just like we discussed, the procaspase 8 molecules are brought close together, they activate each other, and boom, they start activating the executioner caspases.
So an external hit triggers an internal cascade.
Exactly. This pathway, using Fas, is really important for controlling immune cell numbers. If it doesn't work right, you can get autoimmune diseases. Interestingly, cells also have proteins inside that can block DISC formation, acting like a brake or a threshold. Okay, then there's the intrinsic pathway. This one is triggered by signals from inside the cell, often things like developmental cues or cellular stress, like significant DNA damage that can't be repaired. And this pathway hinges on the mitochondria, the cell's powerhouses, doing something quite dramatic. They release specific proteins from their internal space out into the main cell fluid, the cytosol.
And this is where it gets wild, right? Because one of the key players released is cytochrome C. I mean, isn't that normally involved in making energy ATP?
It is. That's his day job in the electron transport chain. But when it escapes the mitochondria into the cytosol during apoptosis, it takes on this completely different deadly role. It moonlights as a death signal.
Incredible.
So once cytochrome C is in the cytosol, it binds to an adapter protein called APAF1. This binding causes APAF1 to change shape and assemble with others into this amazing wheel-like structure, a heptamer 7 units. This wheel structure then recruits the initiator procaspase for this pathway, which is procaspase 9. This whole big complex, the APAF1 wheel plus caspase 9, is called the apoptosome.
The apoptosome.
And just like in the DIC, within the apoptosome, the procaspase 9 molecules are brought together, they activate each other, and then they go on to activate the executioner caspases. This intrinsic or mitochondrial pathway is actually the one responsible for most apoptosis in vertebrates, in us. The absolute key control point is getting those proteins like cytochrome C out of the mitochondria.
Letting them escape.
Yes, through a process called mitochondrial outer membrane permeabilization, or MOMP. And that process is tightly regulated by a really important family of proteins, the BCL2 family.
Ah, the BCL2 family. I've heard of them in relation to cancer.
You absolutely have. They are central regulators. The family is named after BCL2 itself, B-cell lymphoma 2, because it was first discovered in a type of lymphoma where it was overactive, preventing cancer cells from dying. That immediately tells you how important their balance is. So this BCL2 family has basically three main groups of proteins that control that MMP step, the release of cytochrome C. First, you have the anti-apoptotic BCL2 proteins, like BCL2 itself, or another one called BCLXL. These are the survival factors within the family. They sit on the mitochondrial outer membrane and block MOMP. They essentially grab onto and neutralize the proteins that would otherwise poke holes in the membrane.
The good guys, in a sense, for cell survival.
For cell survival, yes. In fact, every normal cell in our body needs at least one of these active to stay alive. Without them, they'd trigger apoptosis. Then second, you have the pro-apoptotic BCL2 family effectors. The main ones are called BAC and BACs. These are the guys that actually do the deed. They form the pores in the mitochondrial outer membrane. When they get an activation signal, they gather together, oligomerize, in the membrane and create the openings that let cytochrome C and other factors escape into the cytosol, triggering the apoptosome formation.
So they're the pore formers.
Exactly. BAC is usually already sitting on the mitochondria, waiting. BACs often floats around in the cytosol until it gets activated. Then it rushes to the mitochondria to help BAC. And the third group, which is really the key link to the signals, are the pro-apoptotic BH3-only proteins. This is a diverse group. BAD, BIM, BID, PUMA, NOXA, lots of them. They are the sensors and messengers. They sense stress signals like DNA damage or receive signals from development, and they promote apoptosis by controlling the other two groups. Well, some of them work by grabbing onto the anti-apoptotic proteins like BCL2.
Pulling them away.
Essentially, yeah. They inhibit the inhibitors. By binding to BCL2 or BCLXL, they free up BAC and MOX to do their pore-forming job. Others in the BH3-only group can actually directly bind to BAC and BAC ends and help activate them.
So these BH3-only proteins connect the upstream stress signals to the downstream pore formers, BACs and BAC. What about that DNA damage example again? How does that link up?
Perfect example. If a cell has DNA damage it just can't fix, this guardian protein, the tumor suppressor P53, becomes active and increases in level. P53 then switches on the genes for some of these BH3-only proteins like PUMA and MOXA. PUMA and NOXA then go and activate the intrinsic pathway, likely by inhibiting the anti-apoptotic BCL2 proteins. This triggers MOMPs, cytochrome seek release, apoptosome formation, caspase activation, and the potentially cancerous cell is eliminated. It's a critical safety mechanism.
Wow.
And there's even more connection. Remember how we said the extrinsic pathway can sometimes recruit the intrinsic one?
Yeah, for amplification.
Exactly. Well, the initiator caspase from the extrinsic pathway, caspase 8, can actually cleave one of those BH3-only proteins called BID. This cleavage activates BID, and active BID then travels to the mitochondria to help activate BAC and BACs, triggering MOMP. It's a direct link between the two pathways.
It really sounds like once these cascades, especially the caspase cascade, get going, it's just full steam ahead. Are there any breaks left at that point? Any final safety checks?
That's a really astute point. Yes, cells do have some further checks and balances, mainly to make sure caspases don't get activated accidentally. One important set of breaks comes from proteins called IAP's inhibitors of apoptosis. Funnily enough, they were first found in viruses. Viruses use them to stop the host cell from killing itself before the virus can replicate. Clever, eh? Yeah. But our cells have their own versions. A key one in mammals is called XIAP. It hangs out in the cytosol and can directly bind to and inhibit both initiator caspase 9 and some of the key executioner caspases like caspase 3. It basically sets an inhibitory threshold. XIAP can even tag these caspases for destruction. But the cell has a countermove. When the intrinsic pathway gets activated and MOMP occurs, the mitochondria release not only cytochrome C, but also a couple of other proteins, Smac and Omi. These are anti-IAP proteins.
Inhibitors of the inhibitor.
Precisely. Smac and Omi bind to XIAP and block it from inhibiting the caspases. So releasing these anti-IAPs helps ensure that once OMP happens, the caspase cascade can really fire properly. This might be another reason why the extrinsic pathway sometimes needs to trigger the intrinsic one to release these anti-IAPs and overcome the IAP breaks.
Okay, so we've got internal death signals, external death signals. What about the opposite? Signals from outside telling a cell, no, don't die, stick around. Survival signal.
Absolutely essential. These are extracellular survival factors. Most cells in multicellular animals like us actually depend on receiving continuous signals from their neighbors to avoid triggering apoptosis, usually via that intrinsic pathway.
So they need constant reassurance.
Kind of, yeah.
Yeah.
It's a way to ensure cells only survive where and when they're actually needed. Think about the developing nervous system. Far more nerve cells are produced initially than are ultimately needed. These neurons then compete for limited amounts of survival factors that are released by the target cells they're trying to connect to. Only the neurons that get enough survival factor survive. The rest, they die by apoptosis.
Wow, so it matches the number of neurons to the size of the target tissue.
Exactly. It's an incredibly elegant mechanism for sculpting tissues. These survival factors bind to receptors on the cell surface, triggering signaling pathways inside the cell that actively suppress the apoptotic machinery. How do they do that? Well, a couple of main ways. Some signaling pathways boost the production of those anti-apoptotic BCL2 family proteins we talked about, like BCL2 or BCLXL, tipping the balance towards survival. Other pathways work by inactivating the pro-apoptotic BH3-only proteins. For instance, a key survival signaling pathway activates an enzyme called ACT kinase. ACT then phosphorylates, adds a phosphate group to a BH3-only protein like BAD. This modification stops BAD from promoting apoptosis, thereby promoting cell survival. So we've gone through the decision and the execution, but let's circle back to the cleanup. You said it's remarkably tidy. How does that happen after the cell breaks into those apoptotic bodies? Right. The cleanup, or phagocytosis, is critical. Those apoptotic bodies, still wrapped in membrane, are quickly recognized and engulfed by neighboring cells or specialized phagocytes like macrophages. This prevents any leakage of cellular contents, which, as we said, would cause inflammation and damage. The key is those eat me signals on the surface of the apoptotic cell. The most well-studied one is a phospholipid called phosphatidylserine, or PS. Normally, in a healthy cell, PS is kept strictly on the inner side of the cell membrane by an enzyme called a flipase.
Hidden away.
Exactly. But during apoptosis, those executioner caspases do two things. They shut down the flipase that keeps PS inside, and they activate another enzyme called a scramblase, which randomly shuffles lipids between the inner and outer layers. The net result is that PS rapidly appears on the outer surface of the apoptotic cell. Phagocytic cells have receptors that specifically recognize this exposed PS, often with the help of bridging molecules, and that triggers engulfment.
Like waving a flag saying, take me away.
Pretty much.
Yeah.
It's also worth noting that healthy cells often display don't eat me signals to prevent accidental engulfment. Apoptotic cells need to lose or inactivate these signals as well to ensure they get cleared efficiently.
This whole balance, life and death at the cellular level, it's just fundamental. So what happens when it goes wrong? When this incredibly precise system gets unbalanced, how does all this molecular detail connect to actual human diseases?
That connection is incredibly direct and, unfortunately, quite common. Problems with apoptosis, either too much or too little, are at the heart of many diseases. If you have excessive apoptosis, too many cells dying, that can cause significant problems. Think about heart attacks or strokes. The initial damage might be necrosis due to lack of oxygen, but in the surrounding area, many cells that initially survive actually die later by apoptosis. This contributes significantly to the overall tissue damage.
So stopping that later wave of apoptosis could potentially help.
That's a major hope, yes. Developing drugs that could block apoptosis in those specific situations might reduce tissue loss after a heart attack or stroke. Then there's the other side. Insufficient apoptosis. Too few cells dying when they should. We mentioned ALPS earlier, the autoimmune lymphoproliferative syndrome, where faulty fast signaling means lymphocytes don't die properly, leading to immune problems. But the really big one here, as you alluded to earlier, is cancer. Evading apoptosis is considered one of the hallmarks of cancer. Cancer cells very often have defects in their apoptotic pathways that allow them to survive signals that would kill a normal cell.
Like the BCL2 example in lymphoma.
Exactly. Overexpression of anti-apoptotic proteins like BCL2 makes those cancer cells resistant to dying. It helps them accumulate, and it also makes them harder to kill with chemotherapy or radiation, because many of those treatments work precisely by inducing apoptosis in cancer cells. And the p53 connection is huge, too. With p53 mutated in roughly half of all human cancers, those cells lose that critical quality control mechanism. Cells with DNA damage don't die. Instead, they survive, potentially accumulate more mutations, and become more aggressive. And again, they become less sensitive to many cancer therapies.
So if blocking apoptosis is bad in cancer, leading to survival, can we flip that? Can we specifically trigger apoptosis in cancer cells as a treatment?
Yes, and that's exactly what some of the most exciting modern cancer therapies aim to do. This has led to the development of drugs called BH3 mimetics.
Mimicking those BH3-only proteins.
Precisely. These are small molecule drugs designed to fit perfectly into the groove on anti-apoptotic BCL2 proteins, where the BH3-only proteins would normally bind. By blocking that groove, they essentially mimic the action of the pro-apoptotic BH3-only proteins. This effectively unleashes back and backs, triggers the intrinsic pathway, and causes the cancer cells to undergo apoptosis. A key example is venetoclax. It's particularly effective in cancers, like certain leukemias, that are highly dependent on specific anti-apoptotic proteins for their survival. It's a fantastic example of how understanding the deep molecular biology leads directly to targeted therapies.
Wow. That really brings it full circle. What an amazing journey. From sculpting fingers and toes to these incredibly sophisticated cancer drugs, all hinging on this process of controlled cell death. It's genuinely awe-inspiring.
It really is. It underscores just how fundamental this balance between life and death is at the cellular level. Understanding these intricate mechanisms, it doesn't just explain biology. It gives us powerful insights into health and disease and potentially new ways to intervene. And it does make you wonder, doesn't it, what other processes in biology that seem on the surface purely destructive might actually turn out to be absolutely essential for life as we know it.
That's a great question to ponder. Thank you for joining us on this deep dive into the fascinating and frankly vital world of apoptosis. We really hope this has given you a new perspective on the hidden complexities working constantly to keep our bodies running. Definitely keep exploring your own curiosities, and we hope you'll join us again next time for another deep dive into the knowledge stacks.